Tonight, we're going to confront an uncomfortable truth about the greatest dream our species has ever had. We want to explore the stars. We want to cross the void, reach distant suns, find out what's out there in the dark between worlds. And we've convinced ourselves that the main obstacle is the distance, or the speed of our rockets, or the cost of the mission. But the real barrier isn't the technology. It's you. It's the body you're living in right now. The one you've carried your entire life without once thinking about what it needs to survive, because everything has always been right here. It was built for one place and one place only. And that place isn't the stars. By the end of tonight, you're going to understand why the cosmos may be the one frontier where our curiosity outreaches our biology. Not because we lack the will, but because the body we evolved into may be fundamentally incompatible with what lies between us and the stars. Before we get started, if you love exploring the depths of space as much as we do, take a second to like the video or subscribe. It's a simple action, but it helps this channel reach more curious minds like yours. Now, let's begin. There's a version of the future that almost everyone who has looked up at the stars has imagined. Humanity moving between solar systems, ships carrying people through the dark between distant suns, crew members walking under alien skies on worlds no human has ever touched. It's a vision so embedded in our culture, it feels almost inevitable, like it's just a matter of time and ambition and engineering before the galaxy opens up to us. But spend some time with the actual biology, the real numbers from real astronauts in real space, and that vision starts to come apart. Because what decades of human spaceflight have taught us isn't that the stars are waiting for us to develop the right engine. What we've learned is that leaving Earth, even briefly, even just to the altitude of the International Space Station, does something to the human body that we cannot fully stop. Something that accumulates with time. Something that interacts with other changes and compounds them. Something that, beyond a certain threshold of duration and distance, we may not be able to reverse. The human body is not a general-purpose exploration vehicle. It is a machine calibrated over millions of years of evolution to work in one specific environment: Earth's gravity, Earth's atmosphere, Earth's magnetic field, Earth's air pressure, its day-night cycle, its temperature range, its radiation levels, its specific microbial environment. Every system inside you, your skeleton, your heart, your muscles, your immune cells, your vision, your hormones, your kidneys, your gut microbiome, your DNA repair machinery, your circadian clockwork developed against the backdrop of this one specific planet. Strip that backdrop away, and the machine starts to fail. Not immediately, not catastrophically in the first minutes. Gradually, relentlessly, and in ways that interact with each other over time, in ways we are only beginning to map. Begin with gravity, because everything about the structure of your body assumes its presence. You feel it right now, pressing you gently and constantly against whatever surface you're resting on. It's so continuous, so perfectly matched to the biological architecture you were born with, that you don't register it as a force at all. You just experience it as being here, as occupying space. But your body isn't passively tolerating gravity. It is actively responding to it, moment to moment, day to day, and it has been since before you were born. Your skeleton exists, the way it does, because of gravity. Bone is not passive calcium. It is living tissue with its own metabolic life, undergoing constant renewal through a process called remodeling. Old bone is broken down by specialized cells called osteoclasts. New bone is laid down by cells called osteoblasts. The two processes stay in balance because mechanical signals from the skeleton tell the body how much structural material it needs. When you stand, walk, carry weight, or run, stress is transmitted through your bones. Tiny deformations occur. Fluid flows through microscopic channels in the bone matrix. Cells detect these signals, and the remodeling process responds by maintaining or increasing density in the stressed regions. This is why astronauts, athletes, and people who do heavy physical work tend to have denser bones than sedentary people. The body builds what the load demands. Remove the load, and the body draws the logical conclusion. It decides the structural material is unnecessary overhead, and it begins breaking it down. This is not a malfunction. The osteoclasts aren't going rogue. The system is working exactly as it was designed to work. In the absence of mechanical load, the remodeling balance tips toward breakdown. Calcium is released from bone mineral into the bloodstream, transported to the kidneys, and excreted. The bone becomes lighter, less dense, less capable of bearing the loads it evolved to handle. Astronauts on the International Space Station lose bone density at a rate of roughly 1 to 2% per month in the load-bearing regions. The lumbar spine, the hip, the femur. 6 months aboard the station, a standard mission duration costs 10 to 15% of bone mass in those regions. For context, 10 to 15% bone loss in the load-bearing skeleton describes the level of compromise seen in people with diagnosed osteoporosis. Their bones become brittle enough that fracture risk rises sharply. Simple activities that a person with a healthy skeleton takes for granted, getting out of bed, stepping down from a curb, carrying groceries, can result in fractures. And the recovery from spaceflight-induced bone loss is slow, requiring months to years of weight-bearing exercise and, in some cases, medication. Not all the lost bone comes back. Some studies show that even after years of recovery, astronauts who flew long missions haven't fully regained their pre-flight bone density. Some of what was lost stays lost. Now, extend that timeline to what an interstellar mission would require. Even at the most optimistic speeds any propulsion system we can seriously discuss could achieve, reaching a star more than 4 light-years away would take decades. Not months. Decades. Even a Mars mission, which involves a mere 6 to 9 months in microgravity each way, produces bone loss in the transit phases that is a genuine medical concern. For a voyage measured in decades, the bone loss doesn't stop. It continues month after month, year after year. What the skeleton looks like after 20 years in microgravity, we cannot say with certainty, because no human has ever been in microgravity for 20 years. What we can say is that the rate of loss, documented from ISS data, extended across a 20-year timeline, describes a skeleton so structurally compromised it may be unable to function in any gravity environment. A crew that arrives at another star system, unable to stand up, unable to bear their own weight, unable to perform the physical tasks that any landing and planetary exploration would require, has not successfully completed the mission. They have survived the transit in a technical sense. But the body that arrives is not capable of doing what it was sent to do. Muscle follows the same logic as bone, and the consequences compound those of skeletal deterioration. Without the constant demand of supporting your own weight against gravity and moving through a gravitational field, muscle tissue deteriorates rapidly and, specifically, the type 1 slow-twitch fibers used for sustained postural work, the ones that keep you upright hour after hour, atrophy preferentially. Fast-twitch fibers used for explosive movements also weaken, though the pattern of loss differs. The neuromuscular connections between the nervous system and muscle tissue, the junctions that allow precise motor control, degrade in accuracy. Astronauts on 6-month ISS missions lose up to 20% of their muscle mass, despite performing 2.5 hours of structured, supervised exercise every single day. That exercise program isn't optional wellness time. It is a medical countermeasure, prescribed and monitored like medication, designed specifically to slow the inevitable loss. It slows it. It cannot stop it. The gap between what exercise achieves and what a gravity environment achieves is fundamental. Gravity provides a low-intensity, constant, omnidirectional mechanical load on every muscle in the body, every minute of every day, including while you sleep. The muscles that stabilize your spine while you sit in a chair, the ones that make micro-adjustments to your balance every time you shift your weight, the ones that hold your head upright and orient your gaze. They are all working in a gravity environment, constantly, effortlessly in your conscious experience, but continuously and importantly. No exercise protocol replaces that. You cannot do 2.5 hours of resistance training and achieve the same biological stimulus that gravity provides over 24 hours. For a crew on a mission lasting decades, the progressive loss of muscle mass and functional capacity would accumulate across the full voyage. What arrives at the destination is a crew that has been atrophying, despite their best countermeasures, for 20 or 30 or 40 years. Some of them will be chronologically old by the time they arrive, compounding the age-related muscle loss that happens to everyone with the spaceflight-induced loss that happens in microgravity. The combination may produce a crew that is physically incapable of the demands that any planetary landing and surface operations would place on them. The cardiovascular system undergoes changes in microgravity that are less visible than bone and muscle loss, but no less medically significant, and some of them may be harder to reverse. On Earth, your heart performs a specific function every beat that is shaped by the geometry of living in a gravitational field. It must generate enough pressure to push blood upward from the chest to the brain against the pull of the planet. The brain sits roughly 12 to 15 inches above the level of the heart in a standing adult. Maintaining adequate blood flow to the brain at that height, against gravity, requires the left ventricle to generate pressures that shape its muscular architecture. In microgravity, there is no uphill. There is no gravitational gradient pulling blood toward the feet. The hydrostatic pressure difference between the feet and the head, which in a standing person on Earth might be about 100 millimeters of mercury, approaches zero. The cardiovascular system responds to this changed reality over the first days and weeks in space. Blood volume decreases because the body interprets the shift of blood toward the upper body as a state of excess fluid volume and triggers mechanisms to reduce it. Total blood volume falls by roughly 10 to 15% in the first week of spaceflight. The heart, freed from the demand of pushing blood upward against gravity, begins to decondition in exactly the way any muscle deconditions when its specific workload is removed. The left ventricle loses mass. It becomes more spherical in shape. Resting heart rate changes. Blood pressure decreases. The cardiovascular system adapts to its new environment. This is efficient. It is also catastrophic when the environment changes back. When astronauts return from long missions and attempt to stand upright, Their cardiovascular system, optimized over months for the low-demand environment of microgravity, cannot adequately regulate blood pressure against the suddenly reappeared gravitational gradient. Blood pools in the legs. Venous return to the heart falls. Cardiac output drops. Blood pressure in the vessels supplying the brain drops. Astronauts feel lightheaded, dizzy, sometimes extremely unwell. Some faint immediately on landing. Most need assistance walking from the spacecraft. This condition, called orthostatic intolerance, is one of the most consistently documented effects of long-duration spaceflight. And it takes weeks to months to resolve through careful reconditioning. For a mission lasting decades, the cardiovascular adaptation to microgravity would be far deeper, far more complete, and potentially irreversible. A heart that has spent 30 years adapting to microgravity may genuinely lack the structural capacity to sustain adequate cerebral perfusion in Earth's gravitational field. The question of whether a person who spent 30 years in space could successfully return to an Earth-like gravity environment is not one we can answer from existing data. No one has tried. The cardiovascular adaptation studies we have are from missions of 1 to 14 months. Extending that data to multi-decade timescales is extrapolation, not measurement. The extrapolation is not encouraging. The visual system is one of the more recent additions to the list of spaceflight-related medical concerns, discovered not through prediction, but through observation of the astronauts themselves. And it sits near the top of the list of unsolved problems in long-duration human spaceflight. Roughly 60% of crew members who complete long-duration missions aboard the International Space Station experience some change in their visual function. Not all of these changes are severe. Some are subtle shifts in refractive error, slight changes in how sharply the eye focuses. But the underlying mechanism, as best as it is currently understood, involves structural changes that go deeper than a simple prescription adjustment would address. The working hypothesis, supported by MRI imaging studies conducted on astronauts before and after missions, is that in microgravity, the redistribution of fluid toward the upper body raises intracranial pressure, the pressure of the cerebrospinal fluid that surrounds the brain and spinal cord and fills the space around the optic nerves. The optic nerve runs from the back of the eye to the brain, enclosed in a sheath filled with cerebrospinal fluid. When intracranial pressure rises, that pressure is transmitted along the optic nerve sheath and acts on the back of the eyeball. The result, documented in post-mission MRI studies, can include swelling of the optic disc, the place where the nerve enters the eye, flattening of the posterior sclera, the back wall of the eyeball, and changes in the shape of the retinal surface. The focused image that falls on the retina depends on the eye having a specific shape and a specific length. When the shape changes, the focus changes. And for some astronauts, these structural changes persist after return to Earth. The eye doesn't simply spring back to its pre-flight geometry when gravity returns and intracranial pressure normalizes. The tissue has been remodeled. Some astronauts have returned from long missions with permanently reduced visual acuity that has not recovered in the years since their flight. The fraction, experiencing lasting changes, is not trivial. The mechanism is still being studied. And the implications for a mission lasting decades are severe. A crew member who experiences progressive vision loss over 30 years in space may arrive at the destination with vision too impaired to function effectively. Navigation, instrument reading, surface operations, any task that requires the ability to see clearly depends on a visual system that is functioning. And we currently have no reliable way to prevent the changes that accumulate in a significant fraction of people in the microgravity environment. Lower body negative pressure devices, which create a partial vacuum around the lower body to pull fluid downward and potentially reduce intracranial pressure, are being studied as a countermeasure. Some preliminary results are encouraging. But whether they can prevent the long-term structural changes in the eye over multi-decade missions is completely unknown. The problem was only identified in the last decade or so. The countermeasure research is just beginning. The immune system runs through a different set of changes in space. But the direction they trend is not reassuring for anyone who needs to stay healthy in an environment with limited medical backup. White blood cells, the cellular foot soldiers of immune defense, show altered function in microgravity. Natural killer cells, which are the immune system's primary rapid response force for detecting and destroying infected or cancerous cells, show reduced cytotoxicity. Their ability to identify and kill abnormal target cells is measurably impaired. T cells, which coordinate the adaptive immune response and are essential for fighting off viral infections and for immune memory, respond differently to stimulation in microgravity. The overall inflammatory signaling profile of the body shifts in ways that affect multiple aspects of immune function. And the practical result of these functional changes shows up in a specific, observable way. Viruses that normally remain dormant in the body become active at higher rates in space. The varicella zoster virus, which causes chickenpox on initial infection and then remains latent in nerve ganglia for life, is found reactivating in astronauts during and after spaceflight at significantly higher rates than in ground-based comparison populations. Epstein-Barr virus, cytomegalovirus, herpes simplex viruses, members of the herpes family that most adults carry in a dormant state, all show evidence of increased reactivation in the spaceflight environment. Most of the time, these reactivations don't produce severe clinical illness, but they produce measurable viral shedding, increased immune system workload, and a clear signal that the balance between host defenses and latent pathogens has shifted in an unfavorable direction. In a confined spacecraft, where a small crew shares recycled air and common surfaces, a more permissive environment for viral reactivation affects everyone aboard. Wound healing is slower in space. Infection risk is elevated. The implications for a decades-long voyage are not comfortable to work through. Over a 30-year mission, the cumulative probability of a serious infectious event, something that a healthy immune system on Earth would handle, but a space-adapted immune system might not, is significant. And beyond the direct infectious risk, immune dysregulation affects inflammatory processes throughout the body. Chronic, low-grade inflammation, driven by immune signaling that isn't functioning the way it was calibrated to function, contributes to tissue damage, accelerated aging at the cellular level, and potentially to some of the other health changes observed in spaceflight, cardiovascular effects, neurological effects, the changes in gut function. The immune system doesn't operate in isolation. It is woven into the function of every other body system through signaling networks of extraordinary complexity. When it operates differently, the effects propagate. Radiation is the problem that, for any mission beyond Earth's magnetic field, sits at the top of the list of things that cannot be fully solved with current technology. Understanding why requires understanding what the space radiation environment is and what it does to biological tissue. On Earth's surface, you are shielded from the radiation that fills space by two protective structures working in combination. Earth's magnetic field, generated by the circulation of molten metal in the planet's outer core, extends tens of thousands of miles into space in every direction, creating a vast protective bubble called the magnetosphere. Charged particles streaming outward from the sun, the solar wind, are deflected by this field, following curved paths that spiral around field lines and are funneled toward the polar regions. The high-energy charged particles that constitute galactic cosmic rays, atomic nuclei, accelerated to extreme energies by supernova explosions and other violent cosmic events, are also deflected, though not as completely. Earth's atmosphere, the roughly 10 metric tons of air sitting above every square meter of the planet's surface, absorbs the radiation that makes it through the magnetospheric deflection. The combined effect is that a person on Earth's surface receives roughly 0.3 rem of radiation from all natural background sources per year. That is the baseline. That is what life here costs in radiation exposure. On the International Space Station, orbiting at an altitude of about 250 miles, but still well inside the magnetosphere, astronauts receive radiation doses roughly 150 to 200 times higher than people on the ground. A 6-month mission exposes them to approximately 8 to 16 rem. This is weighed against mission objectives, judged acceptable for informed volunteer astronauts, and results in a roughly 1% added lifetime cancer risk per 6-month mission. That's the number in low Earth orbit, with partial magnetic protection. Beyond the magnetosphere, in deep space, which is where any mission beyond the moon necessarily travels, neither shield is present. The solar wind hits the spacecraft directly. Galactic cosmic rays flow through unimpeded. NASA's measurements from the Curiosity rover's transit to Mars, using its radiation monitoring instruments, showed that the radiation dose rate in interplanetary space during that transit was more than 300 times the dose rate on Earth's surface. A round trip to Mars, with transit exposures combined, is estimated to increase crew cancer risk by roughly 5%. 5% added risk on top of whatever the individual's baseline cancer risk already was. That is considered to be at the outer edge of acceptable for a mission of this scope and scientific importance. Mars is the nearest other planet. An interstellar mission, spending not months but years and then decades in deep space, would accumulate radiation dose at those elevated rates continuously. The cancer risk from a 20-year transit to a nearby star system at interplanetary space radiation levels would be not 5%, but potentially several 100% of lifetime risk, which is to say, cancer risk, essentially certain during the voyage. But cancer, as serious as it is not even the most immediate concern raised by the deep space radiation environment. The concern that has emerged most sharply from research over the past 2 decades is what high-energy cosmic rays do to the brain. Galactic cosmic rays include heavy ions, carbon nuclei, oxygen nuclei, iron nuclei, accelerated to speeds that can approach the speed of light. These particles carry kinetic energies that dwarf anything produced in particle accelerators. When they pass through living tissue, they don't stop at a single cell. They travel in straight lines through whatever tissue is in their path, ionizing atoms along a narrow track, shattering molecular bonds, breaking both strands of the DNA double helix simultaneously in the cells they traverse, and creating cascades of secondary radiation in the surrounding tissue. The brain is not a structure that regenerates robustly. Neurons, the cells that carry out every cognitive function, don't replace themselves the way skin cells or blood cells do. When a neuron is damaged beyond repair, it is usually gone. And research exposing animals to heavy ion radiation at doses simulating the space radiation environment has produced results that demand attention. Animals exposed to simulated galactic cosmic ray radiation showed impaired spatial learning and memory. They performed worse on problem-solving tasks. Their behavior changed in ways consistent with increased anxiety. And post-mortem examination of their brain tissue showed structural changes, including reduced density of dendritic spines, the tiny extensions on neurons, where synaptic connections between brain cells form. Fewer synaptic connections means reduced information processing capacity. Reduced information processing capacity means reduced cognitive function. The specific human cognitive functions most sensitive to this kind of damage are the ones that matter most on a complex, high-stakes mission: working memory, decision-making, the ability to handle novel situations, and the integration of information from multiple sources into coherent situational awareness. If a crew's cognitive function degrades significantly over a multi-decade mission due to accumulated cosmic ray exposure, every aspect of mission safety degrades with it. The people who need to catch the error, recognize the abnormal reading, make the judgment call under pressure. Those people are making those decisions with brains that have been working in a heavy ion radiation environment for 20 years. We have no human data on what that means. We have animal data that suggests it matters. Now, consider all of these biological stresses, not one at a time, the way we've been discussing them, but simultaneously. Because that's how they operate. The bone loss and the muscle loss and the cardiovascular deconditioning and the vision changes and the immune suppression and the radiation damage don't take turns. They all happen at the same time, to the same person, continuously, from the moment the spacecraft leaves Earth, and they interact. Radiation suppresses immune function through direct damage to the rapidly dividing cells of the immune system, adding to the immunosuppression from microgravity itself. The result of both operating together is greater immune vulnerability than either would produce alone. Cognitive effects from radiation interact with the psychological stress of confinement to produce compounded mental health burden. Bone loss weakens the skeleton. At the same time, muscle loss reduces the muscular protection that would otherwise buffer the skeleton from impact forces. Fatigue from sleep disruption impairs the motivation and capacity for the daily exercise that partially counters bone and muscle atrophy. Immune dysregulation contributes to systemic inflammation that affects multiple organ systems simultaneously. The body's reserve capacity, its ability to tolerate one stressor because other systems are functioning well and can compensate, erodes as those other systems are themselves degraded. This is compounding damage, not additive, compounding. Each impairment reduces the system's ability to resist the others. The biological resilience that healthy people on Earth have as a buffer against illness, injury, and stress gradually depletes as each subsystem that contributes to that resilience is degraded by the space environment. A crew that is simultaneously dealing with compromised immune function, degraded vision, weakened bones, reduced muscle mass, cardiovascular deconditioning, cognitive impairment from radiation, chronic sleep disruption, and the psychological weight of years in confinement is a crew with very little reserve. Very little margin between the state they're in and the state in which they can no longer function. The medical dimension of this problem is not just a clinical concern, it's an operational one. Because in space, there is no hospital. In deep space, there is no rescue. What the crew has is what's on the ship. The pharmaceutical supplies loaded before launch, in quantities calculated to last the mission duration, with no possibility of resupply. The medical knowledge, distributed among the crew, weighted toward whoever was designated the medical officer. The diagnostic equipment, whose inclusion was negotiated against mass constraints. Some pharmaceuticals degrade over time, losing potency. Medications that require refrigeration must remain refrigerated across decades. Sterile. Supplies don't stay sterile indefinitely. And there is no resupply mission. If the crew exhausted their supply of a critical medication in year 15 of a 40-year voyage, that's the situation for the remaining 25 years. Appendicitis happens. Kidney stones happen. Cardiac events happen. Serious infections happen. Cancer happens, and given the radiation environment of a long space voyage, cancer is more likely, not less. Stroke happens. Traumatic injury from a fall or equipment failure happens. Dental emergencies that become systemic infections if untreated happen. All of these are manageable in a modern hospital setting with appropriate specialists, imaging equipment, surgical facilities, and pharmaceutical support. None of that exists on a spacecraft. The medical officer performs what procedures are possible with what equipment and supplies are on board. The alternative is that the crew member doesn't receive adequate care. For any serious condition requiring capabilities the spacecraft doesn't have, the outcome is determined by what the crew can improvise. Telemedicine can extend the medical capability of an isolated crew by connecting them to specialists on Earth. But at interstellar distances, the communication delay measured in years makes real-time consultation meaningless. By the time a question describing a medical crisis reaches Earth and an answer returns, the crisis resolved years earlier. One way or the other. The medical isolation of a crew on an interstellar mission is total. They are completely self-sufficient, or they are not. And given the range and probability of medical events that can occur in any group of people over decades, the certainty that at least one crew member will face a medical crisis, the mission cannot adequately address approaches, certainty as the mission length extends. The psychological architecture of the human mind is no better suited to the interstellar environment than the physiological architecture of the human body. People are social animals, not in a cultural sense, in a biological one. The human brain evolved in the context of a complex social environment, and its architecture reflects that. The neural systems that regulate emotion, motivation, stress response, and cognitive function were shaped by hundreds of thousands of years of living in groups, in open environments, with variety and novelty and the constant sensory richness of the natural world. None of that exists in a spacecraft. A spacecraft is a closed metal environment of fixed size. The same corridors, the same rooms, the same faces, day after day, for the duration. The same recycled air, the same artificial lighting. No weather, no seasons, no horizon. No sound of wind or water or any other living thing except the people on board. The psychological literature on isolated, confined environments is not sparse. Decades of research from Antarctic stations, from submarine deployments, from extended cave isolation studies, from simulated Mars mission experiments conducted in sealed habitat structures, has characterized the psychological trajectory of humans in confinement with some consistency. The first phase is typically high engagement. The novelty of the experience, the sense of mission, the social cohesion of a new team working together, these sustain motivation and morale. Then it changes. The novelty expires. The same faces become familiar in a way that stops being comforting and starts being confining. Small irritations accumulate. Habits that were tolerable in the first weeks of the mission become, over months, sources of genuine friction. The cognitive space of the group, no longer filled with the variety and stimulation of a normal life, contracts around the irritants. Conflicts emerge, sometimes over real disagreements, often over things that would seem trivial in any other context. Interpersonal dynamics become complicated and fragile. Motivation declines. Sleep worsens. Depression rates increase. In the research literature, this is sometimes called the third quarter phenomenon in missions of fixed duration. The third quarter tends to be the most psychologically difficult, when the early engagement has long faded and the end is still not in sight. The Mars 500 simulation, which locked a crew in a sealed habitat for 520 days to simulate a Mars mission, produced exactly this pattern. Sleep disrupted. Activity levels dropped markedly. Some crew members became deeply sedentary and socially withdrawn. And 520 days is less than 18 months. An interstellar mission lasting 40 years offers no third quarter. There is no fixed endpoint to provide orientation. There is no knowledge that the voyage will conclude in a specific number of months. There is only the ship, the crew, the darkness outside, and the impossibly slow brightening of a destination star that will take decades to resolve into anything more than a slightly brighter point of light. What that does to a mind over 40 years, we don't know. The study hasn't been done. It can't be done without doing it. The first people to actually experience it will be discovering the answer in real time on a mission from which there is no early return if the answer turns out to be something they can't endure. Even the body's timekeeping is a problem in space. The circadian rhythm, the roughly 24-hour biological clock that governs when you sleep and wake, when your cortisol rises in the morning and falls at night, when your core body temperature peaks and troughs, when your immune system runs its maintenance cycles, when your digestive system prepares for a meal, is not a cultural convention. It is written into the genome. Every cell in your body contains molecular clock machinery, genes that activate and suppress each other in rhythmic cycles, encoding time into the biology itself. The master clock in the brain, a small region of the hypothalamus called the suprachiasmatic nucleus, coordinates the body's cellular clocks through hormone signals and neural output. And it is synchronized to Earth's day-night cycle through light signals from the retina. Every morning, as light levels rise, signals from your eyes reset the master clock. The body's time is aligned to the planet's rotation. In space, there is no planet rotating. There is no sunrise. The light environment inside a spacecraft is whatever the lighting system provides. Space agencies work hard to program artificial lighting schedules that support circadian alignment, including adjusting light color temperature and intensity to mimic the cues that reset biological clocks. But the artificial cues are not the same as the real cues. And in a spacecraft traveling through the interstellar dark, there is no external reference. The sun itself, billions of miles behind the ship, has no rhythmic relationship to the crew's experience. Astronauts on the ISS consistently show worse sleep than on Earth. Average sleep duration falls short of recommendations. Sleep quality is degraded. Use of sleep medications is common. Fatigue accumulates. Over a mission of months, this is a manageable challenge. Over a mission of years or decades, chronic sleep disruption would become a permanent feature of the crew's biological reality. Chronically disrupted sleep impairs attention, working memory, reaction time, decision-making quality, emotional regulation, immune function, and metabolic health. It contributes to accelerated biological aging. It interacts with the radiation effects and the psychological stress to compound the burden on the crew. The circadian system is not a luxury feature. It is the timing infrastructure of the entire biological body. When it runs poorly, everything it schedules runs poorly. At the level of individual cells, the space environment is doing things that researchers have only begun to characterize. The twin study that NASA conducted, following astronaut Scott Kelly through a year-long ISS mission while his identical twin Mark Kelly remained on the ground, provided the closest thing to a controlled experiment on the effects of long-duration spaceflight on human biology, because the twins share genetic starting points. What the study found was surprising in its scope. Gene expression, which genes are active, and to what degree changed in Scott Kelly during the mission, in ways that were not seen in Mark Kelly on the ground. Thousands of genes showed altered expression patterns. Most returned to near baseline after the mission ended, but a subset did not. About 7% of the genes whose expression changed during the mission had not returned to baseline levels 6 months after Scott Kelly's return to Earth. Some of the altered gene expression patterns were in pathways related to DNA repair, immune function, bone formation, and the extracellular matrix, the structural scaffolding that holds tissues together. Telomere length, which is associated with cellular aging and the remaining replicative capacity of cells, changed during the mission in a way that initially appeared counterintuitive. Telomeres lengthened slightly in space, then rapidly shortened after return to Earth, ending up shorter than they were before the mission. The long-term significance of these telomere changes is not fully understood. What they indicate is that spaceflight is doing something to cellular biology that isn't simply accelerating the normal aging process. It's producing a different biological trajectory, one whose long-term health consequences we cannot predict with confidence from short-duration mission data. The gut microbiome changed substantially over the course of Scott Kelly's year-long mission. The composition of his intestinal microbial community, the relative abundances of different bacterial species, shifted significantly in ways that weren't seen in his twin brother. Most, but not all, of these changes reverted after his return. The gut microbiome matters because it is not a passive tenant. It influences immune signaling, inflammatory status, metabolic function, and through the gut-brain axis, aspects of cognitive and emotional function. A microbiome that has been substantially reorganized by the space environment is one whose downstream effects on every system it influences have also been reorganized. What that means over a 40-year mission, with the microbiome adapting further and further from the Earth baseline configuration it started with, is unknown. We don't have the data. The year-long ISS mission is the longest direct observation we have. Everything beyond that is extrapolation. And extrapolation carries increasing uncertainty the further from the data it runs. There are proposed solutions to these problems, and they deserve serious engagement rather than dismissal. The researchers working on them are not naive optimists. They are scientists confronting a real engineering and biological challenge with rigorous tools. The first category of proposals involves countermeasures, interventions designed to slow or prevent the biological changes caused by spaceflight without altering the fundamental environment. Pharmacological interventions for bone loss are an active research area. Bisphosphonates, the class of drugs used to treat osteoporosis on Earth, work by inhibiting osteoclast activity, slowing the breakdown of existing bone. Used in combination with exercise, they have shown some benefit in reducing spaceflight-induced bone loss. Newer agents targeting specific signaling pathways involved in microgravity-related bone resorption are under investigation. Sclerostin inhibitors, which block a protein that normally suppresses bone formation, have shown bone-building effects in ground-based osteoporosis treatment and are being considered for space applications. None of these are sufficient to prevent all bone loss across a multi-year mission. They slow the rate. They reduce the total deficit. They don't solve the problem. Pharmacological approaches for radiation protection are also under study. Antioxidants can reduce some forms of radiation-induced oxidative damage. Certain compounds that promote DNA repair activity are being explored. Drugs that modify the inflammatory response to radiation-induced damage may reduce some of the secondary tissue damage. Radioprotective agents that can be taken before anticipated high-dose exposure may provide some benefit during solar particle events. But the problem of chronic, multi-decade exposure to galactic cosmic rays at a level that causes cumulative neurological and oncogenic damage is not addressed by any currently available pharmacological approach. The heavy ion component of galactic cosmic radiation is, with existing materials, essentially impossible to shield against completely. The second category of proposed solutions involves structural changes to the spacecraft environment to better replicate the biological conditions of Earth. Artificial gravity, created by rotating the spacecraft, or a module of it, is the most important of these. The physics is well established. Rotator structure and the centripetal acceleration experienced by objects on the inner surface of the rotation replicates the effect of gravity. Stand on the inner surface of a rotating ring, and you experience a downward force indistinguishable in its biological effects from the force of a gravitational field of the same magnitude. Your skeleton loads. Your cardiovascular system works against the apparent gravitational gradient. The fluid redistribution toward the head doesn't occur. The circadian light environment can be programmed to provide a 24-hour cycle. Artificial gravity would address multiple problems simultaneously: bone loss, muscle atrophy, cardiovascular deconditioning, the vision problem from fluid redistribution. The engineering challenge is real but not impossible in principle. The problem is scale. To produce comfortable, functional, artificial gravity without the disorienting Coriolis effects that make small, fast-rotating structures unusable, you need a large radius. At a radius of a few hundred meters, the rotation rate required to produce 1 g is slow enough that the Coriolis effect becomes tolerable. A habitat ring with a radius of around 250 meters, rotating at roughly 2 revolutions per minute, produces close to 1 g at the rim. That structure would be larger than any human-made object ever placed in space. It would need to be built and launched from Earth, which means lifting its mass out of Earth's gravity well. It would need to be structurally sound enough to maintain its integrity under continuous rotation for decades in the vacuum and radiation of interstellar space. It would need to be pressurized, habitable, and integrated with a propulsion system capable of accelerating the entire assembly, habitat ring included, to a meaningful fraction of the speed of light. The mass of a large rotating habitat competes directly with the mass of the fuel and propulsion hardware needed to reach interstellar speeds. Adding tens of thousands of metric tons of rotating structure to an already mass-critical vehicle is not a free move. It makes the propulsion problem harder, in exactly the situations where the propulsion problem is already at the boundary of what physics permits. Artificial gravity is conceivable. It is not currently achievable for an interstellar mission. Whether it will become achievable within the century is genuinely uncertain. The third category of proposed solutions involves changing not the environment, but the biological system itself. If the human body is poorly adapted to the space environment, one approach is to make it better adapted. This is the domain of genetic and biological enhancement. It is also the most controversial and the furthest from current capability. The concept is that gene therapy or other biological interventions could modify specific aspects of human physiology to make it more tolerant of the space environment. Targets might include the DNA repair pathway to improve radiation resistance, the bone metabolism pathway to prevent microgravity-induced resorption, the intracranial pressure regulatory system to prevent the fluid dynamics that impair vision, the immune system architecture to maintain better function in the altered conditions of space. Biological precedent exists for enhanced radiation resistance. The bacterium Deinococcus radiodurans survives radiation doses roughly 5,000 times the lethal dose for a human. Through a DNA repair mechanism so efficient it can reassemble a genome shattered into hundreds of fragments. The mechanisms responsible for this capability are understood at the molecular level. The genes responsible have been sequenced and characterized. Whether their function could be incorporated into human biology, whether the relevant repair enzymes could be introduced into human cells in a way that enhanced radiation resistance without disrupting. The enormous complexity of human cell biology is not a simple question. Gene therapy has advanced rapidly over the past decade. Tools like CRISPR have made precise genomic editing possible in ways that weren't feasible 20 years ago. Applications in treating single gene disorders have produced genuine clinical successes. But the gap between treating a disease caused by a single defective gene and enhancing the radiation resistance of every cell in a human body is enormous. The number of genetic changes involved, the need to avoid disrupting existing gene networks, the delivery challenges of reaching every relevant tissue, the ethical questions about altering the human germline versus somatic cells only, the regulatory framework that would govern any such enhancement. None of these are simple. None of them are close to resolution. But the direction of travel in biological science suggests that what seems impossible today often becomes possible within a generation or two. The question for interstellar travel is whether that generation is 1 or 5 or 20, and whether humanity will make the social and political choices to pursue it. The fourth path being seriously discussed involves not sending humans at all, at least not first. Robotic and autonomous probes face none of the biological constraints of a crewed mission. An instrument package doesn't lose bone density. It doesn't accumulate radiation damage in a way that impairs function. It doesn't develop psychological problems from isolation. It doesn't require food, water, air, or medical care. A probe can be designed to operate for decades in the deep space radiation environment because silicon and metal and composite materials tolerate cosmic rays in ways that neurons and bone and blood simply cannot. The breakthrough starshot concept, which has attracted serious scientific and financial commitment, proposes sending gram-scale spacecraft on reflective sails, accelerated to roughly 20% of the speed of light by arrays of powerful ground-based lasers. At that speed, a probe could reach Alpha Centauri in approximately 20 years and transmit data back, with the signal taking 4.24 years to arrive. The scientific return from direct observation of another star system would be extraordinary. Atmospheric composition of any planets. Thermal emission data. Magnetic field structure. Whatever the miniaturized instrument package could detect. No crew. No biological problems. Just information arriving back at Earth roughly 24 years after launch. This represents a realistic near-term path toward actual data about nearby star systems, rather than the inferences we currently draw from distant observation. And in the history of exploration, information has always preceded presence. The ocean floor was mapped before it was visited. The lunar surface was photographed in detail before it was walked on. The atmosphere of Mars was characterized before any craft landed on its surface. Applying that pattern to interstellar space suggests that the first steps toward the stars will be informational probes sending back data that tells us what's actually there. The physical steps, if they ever come, will be built on that information, and perhaps informed by a much clearer understanding of what a crewed mission would actually face, having observed the environment directly. There's also the generation ship concept, which takes a completely different approach to the biological problem. Rather than solving the problem of keeping a crew alive across multi-decade transits, it accepts that the crew who launches will not survive to arrive, and designs the mission for the descendants of the launching crew to complete. A vessel large enough to house a self-sustaining community, carrying everything needed to maintain human life and population across centuries of travel, could in principle transport humanity to another star without requiring any individual to survive the full journey. The biological constraints apply to individuals. They don't apply to a population that renews itself biologically along the way. The problems shift from the physiological to the social, political, genetic, and psychological. How large does the founding population need to be to maintain sufficient genetic diversity across centuries without inbreeding depression, degrading the gene pool? Estimates vary, but population geneticists generally suggest several hundred individuals as a minimum for long-term genetic health, with some analyses suggesting populations of 1,000 or more provide better margins. A vessel housing and sustaining 1,000 people for centuries is not a spacecraft in any sense we currently build them. It is a world. A closed, self-sustaining ecosystem with its own agriculture, its own manufacturing, its own governance, its own culture, its own legal and social institutions. Building it and launching it would require resources and engineering capabilities that don't currently exist. And the social dynamics of the mission introduce their own profound uncertainties. People born on the ship didn't choose the mission. They were born into it. The generations who live and complete their lives in the middle centuries of the voyage will have no connection to Earth except as history, and no experience of the destination except as an abstraction described in records. Their psychological relationship to the mission is completely unlike the astronauts who signed up for it. Whether they'll maintain the cultural continuity, the technical competence, the social cohesion, and the institutional knowledge needed to complete the mission across centuries of generations is not a guaranteed outcome. History on Earth offers examples of isolated communities that maintain themselves across generations. It also offers examples of communities that fragmented, lost knowledge, developed internal conflicts that tore them apart, or simply drifted away from the purposes that originally organized them. A generation ship is the largest, most consequential, most irreversible experiment in isolated community dynamics ever attempted. The first one would be discovering in real time whether humanity is capable of it. What all of this adds up to is a picture of the human body as the ultimate bottleneck in our reach toward the stars. Not the distance, not the fuel, not the time, the body. Because the body is the one element that every interstellar mission concept must eventually account for, and the one element that resists the engineering approaches that have solved every other challenge we've confronted in spaceflight. The distance is a physics problem. Physics is hard, but it responds to careful thinking and accumulated knowledge. The propulsion challenge is an engineering problem. Engineering is enormously difficult, but it is a domain where human ingenuity has repeatedly surprised us. But the body is a biological problem, and biological problems don't yield to ingenuity the way engineering problems do. They yield to understanding. And understanding biology at the depth required to reliably keep a human being healthy across decades of conditions it never evolved for is a different category of challenge. It requires mapping systems of complexity that dwarf any spacecraft we've ever designed. It requires developing interventions that work reliably across the enormous individual variation that human biology exhibits. It requires doing all of this safely, in a context where the consequences of getting it wrong are not a mission failure, but a human tragedy. The engineering approach to any of these individual problems leads back to the same place. The body that needs to be protected is more complicated than the protection systems we're designing. The skeleton doesn't respond to exercise countermeasures the way a metal structure responds to reinforcement. The brain doesn't respond to radiation shielding the way electronics respond to a Faraday cage. The mind doesn't respond to social programming the way software responds to updated code. These are biological systems shaped by evolution, operating through mechanisms we've been trying to understand for decades. Resisting simplification, because their complexity was the point. It was what allowed them to thrive in the specific, rich environment of this planet. Remove them from that environment, and they fail in ways that are not always predictable and not always reversible. And yet, the fact that we've identified these problems with the precision we now have is not a reason for despair. It's a form of progress in itself. We don't talk about the biological barriers to interstellar travel the way science fiction talked about the unknown of space 50 years ago, as vague, romantic dangers. We talk about them the way physicians talk about diseases, with specific mechanisms, measured rates, documented outcomes, and research programs aimed at each identified problem. We know the bone loss rate is 1 to 2% per month in load-bearing regions. We know the radiation dose on an interplanetary transit, and we can model it for longer voyages. We know which aspects of vision change and which mechanism drives those changes. We know what immune cell types are most affected in microgravity and how their function shifts. We know that cognitive function is affected by cosmic ray exposure and which brain regions are most vulnerable. We know the psychological trajectory of isolated, confined groups and which phases of that trajectory are most dangerous. We know gene expression changes across hundreds of pathways during spaceflight, and some of those changes persist after return. That knowledge was earned. It came from instrumented missions, from biological samples carefully preserved and analyzed, from animal experiments, from twin studies, from isolated environment analogues on Earth. It reflects decades of dedicated scientific effort by people who understood that human spaceflight, beyond Earth's protection, requires understanding the biology of the traveler as rigorously as the proof. Physics of the journey. And what you can measure, you can eventually address. Not quickly, not easily. But the trajectory of biomedical science, directed specifically at the challenges of long-duration spaceflight, suggests that the problems will look very different in 100 years than they look today. Pharmaceutical interventions will improve. The genetic toolbox will expand. Our understanding of the gut microbiome and how to maintain it will deepen. Sleep medicine will advance. Radiation biology will yield more insights into how to protect the central nervous system. Psychology will develop better models of what long-duration crews need and how to support them. None of this is certain. None of it is on a timeline that makes interstellar travel something the people living today will see. But the direction is toward understanding, and understanding is the precondition for everything else. What the biology of spaceflight ultimately tells us is something profound about what we are, more profound than it might first appear. We are Earth, not in a poetic sense, in a literal biological sense. The carbon atoms in your cells were cycled through this planet's biosphere. The calcium in your bones was deposited through mineral processes specific to this world. The iron in your hemoglobin traces back to the composition of the solar nebula from which this planet condensed, incorporated into life that evolved on its surface over billions of years. Your metabolic rate, the temperature at which your enzymes function optimally, the oxygen partial pressure your lungs are tuned for, the gravitational loading your skeleton requires none of these are arbitrary parameters. They are the output of billions of years of evolution operating in one specific environment. Your circadian clock runs on 24 hours because this planet rotates in 24 hours, and your ancestors' fitness over millions of generations was shaped by that cycle. Your bones are built for 1G because Earth's surface gravity is 1G, and every skeleton that couldn't handle that load didn't pass on its genes. You are not a visitor to Earth. You are an expression of it, a physical consequence of 4 billion years of this planet's specific conditions acting on the chemistry of life. And when you leave those conditions behind, the system starts to fail. Not because space is trying to kill you, though it is, but because the further you travel from the conditions that built you, the more completely those conditions are absent, and the more completely your body's deep assumptions about where it exists turn out to be wrong. The human body assumes gravity. Space provides none. The body assumes a magnetic shield. Space provides none. The body assumes Earth's radiation environment. Space provides something far more hostile. The body assumes a 24-hour cycle of light and dark. Space provides perpetual darkness, punctuated by a star too distant to change. The body assumes a social community of dozens to hundreds of individuals in a complex, varied physical world. Space provides a handful of people in a metal cylinder of fixed dimensions. Every assumption is violated. Every adaptive system is working in conditions it was never built for. And the result, accumulating over months, is damage. Over years, it is serious damage. Over decades, it is damage of a kind and a magnitude that we cannot yet reliably prevent or reverse. This is the reality behind the dream of exploring the stars. Not a reason to abandon the dream. The drive to go, the restlessness, the insistence that the horizon isn't the end that's not going away. It never has. What the biology demands is that the dream be honest. That the technical plans for interstellar missions treat the human body not as a box to be shipped between destinations, but as a complex biological system with specific requirements that don't disappear because the mission needs them to. That the research investment in space medicine be commensurate with the ambition of the missions being planned. That the biological problems receive the same systematic, rigorous, funded scientific attention that the engineering problems receive. That we be honest about the gap between where we are and where we need to be. Because the gap is real. The human body, in its current form, is not ready for the stars. Not the stars that matter, not the ones that would take decades to reach. Not if we need the crew to arrive capable. The timeline for developing the biological solutions is long, possibly as long as the development of the propulsion systems, possibly longer. And that timeline runs parallel to everything else we need to figure out. There is no shortcut past the biology. There is only through it. There is one more dimension of the biological problem that almost never appears in the popular conversation about interstellar travel, and it may be among the most important: the human reproductive system. If a mission is long enough that the crew might want to have children during the voyage, or if the mission design assumes biological reproduction as a strategy for maintaining crew size, genetic diversity, or social stability across decades, then the space environment's effects on human reproduction become a central concern. We know almost nothing about human reproduction in space. Zero. Not because nobody thinks it matters, because we've never studied it. No human pregnancy has ever been attempted or completed in space. No birth has occurred in orbit. This isn't an oversight. It's a consequence of how spaceflight missions have been structured, short enough and staffed with enough careful crew selection that the question never arose operationally. But for a multi-decade interstellar voyage, it arises immediately. What we do know comes from animal studies in microgravity and from the general biological plausibility of what spaceflight conditions would do to the reproductive process. The news is not encouraging. Fertilization and early embryonic development appear to be affected by microgravity in rodent studies. The earliest stages of cell division, which establish the architecture of the developing embryo, depend on oriented structures within the egg cell that may behave differently without the reference of gravity. Studies in orbiting spacecraft have shown disrupted development in sea urchin and frog embryos exposed to microgravity. The human embryo is more complex, but there's no reason to think it's immune to the same mechanical and signaling effects. Implantation and placental development involve the invasion of maternal tissue by embryonic cells in a process that is partially governed by physical forces. Whether those forces operate normally in microgravity is unknown. The cardiovascular changes of spaceflight, the reduced blood volume, and altered pressure dynamics could affect placental blood flow in ways that impair fetal development. The immune changes of spaceflight matter enormously during pregnancy, because maintaining a pregnancy requires specific immune tolerance of the developing fetus. Altered immune function could disrupt that tolerance. Radiation exposure during pregnancy is a serious concern. The developing fetus, with its rapidly dividing cells constructing the entire architecture of a new body, is one of the most radiosensitive biological states that exists. Doses of radiation that might be manageable for an adult can cause severe developmental abnormalities in a fetus, depending on the developmental stage at the time of exposure. During pregnancy in deep space, the fetus would be exposed to the same galactic cosmic ray environment as the rest of the crew, with no additional shielding beyond the mother's body. The risks of developmental abnormalities, miscarriage, or stillbirth under those conditions are genuinely unknown because the study hasn't been done and cannot be done ethically. Any child born on an interstellar voyage would have spent their entire gestation in the space radiation environment and in microgravity, then grown up in the same environment. The developmental effects of spending childhood in microgravity, with its bone metabolism changes, its fluid redistribution, its immune alterations, are completely unstudied. A child who grows up in space is not simply a smaller adult in space. A child's skeleton is forming. A child's cardiovascular system is developing. A child's immune system is maturing. A child's brain is undergoing critical developmental periods during which environmental input shapes architecture that will persist for life. We have no idea what spaceflight conditions do to any of those developmental processes. The concerning answer, extrapolating from what we know about the individual effects of microgravity and radiation on adult systems, is that the effects on developing systems might be more severe, not less. A generation ship whose founding design relies on children being born and growing up on board is betting the biological welfare of those children on assumptions about developmental biology in space that have not been tested and cannot be tested before the mission launches. Those children didn't choose the mission. They were born into it. Whatever the space environment does to their developing bodies is something they will carry for life. If serious developmental problems emerge, there is no medical system adequate to address them. There is no way to send them back to Earth. There is no way to undo the biological consequences of a childhood spent in an environment that human development never encountered before. This is one of the most ethically uncomfortable aspects of any long-duration mission concept that involves reproduction or children, and it gets far less attention than the propulsion systems and the navigation math. The question of what the human body does to time, what happens biologically to a person who spends decades of their life in space, raises a related issue that is rarely framed this way: aging. The human body ages. This is not something that spaceflight changes in the sense of introducing a new process. Aging is already happening. What spaceflight may change is the rate at which various aspects of it occur, and possibly the specific pathways through which it manifests. A crew, launched in their mid-30s toward a destination 40 years away, would arrive, at the earliest, in their mid-70s. They would have spent their entire adult working lives on a spacecraft. They would arrive at the biological age of elderly people after 40 years in an environment that we know accelerates some aging mechanisms. The telomere shortening, documented in the twin study. The altered gene expression patterns that persisted after return. The radiation damage accumulating in every cell. The cardiovascular deconditioning. The musculoskeletal deterioration. In ordinary aging on Earth, these processes unfold against a background of Earth's supportive environment, adequate gravity, appropriate radiation shielding, varied diet, rich social environment, medical care, which provides the body with its best conditions for dealing with the inevitable decline. In space, all of those supportive conditions are absent. The aging processes that Earth's environment would buffer and slow are instead occurring in an environment that actively accelerates some of them. A crew member, at 40 years into a deep space voyage, may be biologically older than their chronological age suggests, having spent 4 decades without the conditions that normally allow the body to maintain itself. The medical needs of this crew upon arrival, even if everything went well, would be those of an elderly population that has spent 4 decades in unusual and physiologically challenging circumstances. They would arrive needing the kind of medical support that currently only exists on Earth. And at their destination, there is nothing. No hospital. No medical infrastructure of any kind. Whatever medical support they need would have to come from what they brought with them, in whatever condition it remained after 40 years. The maintenance and reliability of medical equipment across multi-decade timescales is another challenge that barely gets mentioned in the engineering discussions. Electronic components degrade. Calibration drifts. Moving parts wear. Consumables expire. A dialysis machine that was state-of-the-art when the ship launched may not be functional 40 years later without replacement parts that aren't available. Diagnostic software written in the year of launch may be inadequate for conditions that develop in ways the software wasn't trained to recognize. Pharmaceutical supplies that were loaded for a crew of 20-35-year-olds may be inadequate for a crew of 20-75-year-olds, with the accumulated health burden of 4 decades in space. Every element of the medical system that seemed adequate at launch becomes less adequate as the voyage extends, the crew ages, and the equipment ages. The gap between what the crew needs and what they have access to only grows. There is something important to say about the psychological weight of knowing all of this. For anyone who wants to explore the stars, and the number of people who do, deeply and genuinely, is not small. What we've been discussing isn't an abstract set of scientific concerns. It is a personal confrontation with limits. The limit on the reach of one of the most powerful drives human beings experience. The drive to go, to see what's there, to push beyond the known. Every human culture that has ever existed has expressed this drive in some form. Migration across continents, boats into uncharted ocean, expeditions into polar darkness, descents into the deep sea. Every time, there were people who thought the limit was permanent, and people who pushed against it anyway, and often enough the people who pushed prevailed. The limit on human spaceflight is different from those historical limits in one fundamental way. All previous limits were external. The ocean was wide. The mountain was high. The darkness was cold. But the explorer's body, however stressed by the journey, was built for the planet on which the journey occurred. The physiology of a sailor crossing the Pacific was calibrated for Earth. The same gravity, the same radiation environment, the same atmospheric pressure. The crossing was difficult. It was dangerous. People were lost. But the body wasn't working against the physics of the environment in a fundamental way. It was being stressed by the journey, not undermined by the destination's basic physics. Space is different. The barrier to interstellar exploration isn't the external environment of some unmapped territory. It is the biological architecture of the explorer itself. And that architecture cannot be changed by courage or determination or the sheer force of human will. It can only be changed by understanding it deeply and precisely, and then doing the biology necessary to modify what needs to be modified, protect what needs to be protected, and extend what needs to be extended. Courage is required. But courage alone won't preserve bone density in microgravity. Determination won't shield neurons from galactic cosmic rays. Will cannot replenish a pharmaceutical supply that ran out 15 years from the destination. What's required is science. Real, patient, funded, multi-generational science directed specifically at the human body's incompatibility with the space environment. The interstellar missions of the future, if they ever happen, will have been made possible by research conducted in hospitals and laboratories and space stations over the decades and centuries before the ships were built. The astronauts of those missions will benefit from biological knowledge that doesn't exist yet, from interventions that haven't been developed yet, from a scientific understanding of what the space environment does to the human body that is far deeper and more comprehensive than what we have now. The biology has to come first, not as an afterthought, not as a secondary concern after the propulsion and the navigation and the structure, as a primary constraint given equal weight to the engineering. Because an interstellar ship without a biological solution for the crew is not a mission. It is a tomb. The honest accounting of what we know leads to a conclusion that the optimistic narratives about human spaceflight often avoid. We are not ready. Not in the sense of lacking the right rocket, in the sense of lacking the fundamental biological knowledge and intervention capability needed to keep a crew healthy across the timescales that interstellar travel requires. The nearest star system is 4.24 light-years away. At 5% of the speed of light, an impressive velocity by any current engineering standard, the one-way transit takes 85 years. At 10% of the speed of light, still far beyond what any near-term propulsion technology can achieve, it takes 42 years. Those timescales, 40 to 80 years in deep space, are not biologically survivable with current countermeasures. They are not survivable with any countermeasures we can see clearly on the research horizon. They might become survivable with biological interventions that require decades of research to develop, test, and validate. Some of those research programs are running. Space medicine is a real field, with serious scientists doing important work. The ISS has served as an invaluable laboratory for understanding what spaceflight does to the human body, and the data it has generated has directly shaped our understanding of the problems. But the scale of the research investment relative to the scale of the challenge is not well matched. The amount of money, time, and scientific attention devoted to propulsion concepts for hypothetical interstellar missions substantially exceeds the amount devoted to understanding how to keep a human being biologically functional for the duration of such a mission. That imbalance reflects a broader cultural tendency in the space exploration conversation to be captured by the romance of the destination and the engineering challenge of the journey, and to treat the biological problem as something that will sort itself out. It will not sort itself out. The biology doesn't solve itself because the mission needs it to. It responds to investment, to research, to the accumulated understanding of a scientific community that takes it seriously. There's a perspective on all of this that's worth holding alongside the challenge. The same species that is limited by its biology is the one that identified those limits, that characterized them, that measured them with instruments it designed and built, that connected the bone loss to specific cellular pathways. That traced the vision, changes to fluid dynamics and intracranial pressure. That linked the cognitive effects to the track structure of heavy ion radiation passing through brain tissue. That is not an accident. It is exactly the kind of analytical capability that makes solving hard biological problems possible. You cannot address a problem you haven't identified. You cannot design an intervention for a mechanism you haven't characterized. You cannot protect against damage whose pathway you haven't mapped. We have done the identification. We are doing the characterization. The mapping is incomplete but advancing. The gap between where we are and where we need to be is large, but it is not infinitely large. It is a specific gap between current biological understanding and the understanding required to keep a human body healthy in deep space for decades. That gap is the subject of an active field of scientific inquiry. It will not close in 10 years. It may close in 100. The timescale is geological by human standards of impatience. It is brief by the standards of scientific revolutions, the germ theory of disease, the discovery of antibiotics, the characterization of the human genome. These weren't fast. They required generations of researchers building on each other's work. What they required that time produced is the ability to do things that were previously impossible, to cure infections that were previously fatal, to understand genetic disease at the molecular level, to design drugs that target specific proteins with precision that previous generations couldn't have conceptualized. The biological requirements of interstellar travel are an equally hard problem. They will require equally sustained, equally serious scientific effort. And they will require the same honest acknowledgement that the problem is harder than the optimistic narrative suggests, and the same insistence on doing the work anyway. The stars are extraordinary. The closest one, Proxima Centauri, a red dwarf 4.24 light-years from the Sun, is known to have at least one planet in its habitable zone. Proxima Centauri b, roughly Earth mass, orbiting in a region where liquid water might exist. Whether it does, whether that planet has an atmosphere, whether something lives there. These questions are not answerable from Earth with current instruments. They require going, or at minimum, sending something capable of direct observation from close range. The Alpha Centauri system, just slightly further at 4.37 light-years, is a binary star system of two sun-like stars that have been orbiting each other for billions of years. What worlds exist around them? What they look like? What their environments are? We don't know. The desire to know is not irrational. It is one of the most characteristically human impulses there is. The drive to understand, to see for ourselves, to go and find out rather than accept the limits of remote observation. It's what has driven every expansion of the human known world. And the stars represent the next horizon of that impulse. The desire is real. The drive is real. The dream has weight and substance and deserves to be taken seriously. What it doesn't deserve is to be pursued in a way that ignores the biological reality of what it would require. A mission that sends people to the stars without solving the biological problems isn't bold exploration. It's sending people somewhere to fail in ways that were predictable, preventable, and simply not addressed because the engineering was more interesting to work on. The people of the future who look back on the early 21st century as the era when humanity first seriously contemplated interstellar travel, will see either the generation that was honest about the biology and invested in solving it, or the generation that dreamed about the stars while underinvesting in the science that would have made it possible. The choice about which of those is true is being made now, in funding decisions and research priorities, and the cultural conversation about what space exploration actually requires. There is a specific failure mode in how humanity talks about space exploration that the biology makes visible, and it's one that matters beyond the narrow question of interstellar travel. We treat the human body as the least interesting variable in the equation. The propulsion systems are fascinating. The spacecraft designs are fascinating. The orbital mechanics are beautiful mathematics. The destinations, those distant suns with their unknown planetary systems, are the subject of an entire field of astronomy, generating constant new discoveries. But the body that would have to inhabit the spacecraft, travel in the radiation environment, land on the alien world, and actually do something useful there, the body gets treated as a given, a fixed parameter, something assumed rather than designed for. This shows up in the way we talk about the timeline. Engineering discussions about interstellar propulsion concepts talk about a horizon of 50 to 100 years for when certain technologies might become feasible. Biological discussions about the countermeasures needed for multi-decade spaceflight are rarely put on any timeline at all, because the problems are harder to frame as engineering problems. They're not engineering problems. They're biology problems. And biology doesn't respond to engineering timelines. This mismatch needs to be addressed before any serious interstellar mission planning can be done. Because what good is a ship that can reach another star if the crew arrives physiologically incapable of doing what they were sent to do? The vehicle is not the mission. The people are. And the people need to be the primary design constraint, not the afterthought. The field of space medicine has made genuine progress over the past 3 decades. The data from the ISS has produced a research literature of thousands of papers characterizing in detail what happens to the human body in spaceflight. Some of the findings were expected. Many were not. The vision problem wasn't anticipated in early spaceflight planning. The extent and persistence of bone loss wasn't fully appreciated until systematic measurements were made. The immune system changes weren't fully characterized until sufficiently sensitive assays were applied to samples from returning astronauts. The gene expression changes across hundreds of pathways only became visible when genomic analysis tools became powerful enough to detect them. Each surprise in the space medicine literature is both a warning and an opportunity. A warning because it reveals a problem that wasn't previously counted in the risk ledger. An opportunity because a characterized problem is an addressable problem. The next decade of ISS research, and the research that will come from the Lunar Gateway and from any lunar surface missions that sustain crew members for extended periods, will add to that literature. They will reveal more surprises, more mechanisms that weren't anticipated, more interactions between stressors that weren't predicted from studying each stressor in isolation. And each of those revelations will either need to be addressed or accepted as a limitation on what kind of missions are possible. The pace of revelation matters. The more we learn in low Earth orbit and in the near space environment before attempting anything further, the less we will be surprised by when the further missions actually happen. This is the argument for a conservative, step-by-step approach to human expansion into the solar system. Not because the dream should be constrained, but because each step teaches us something about the biology that the next step depends on. A lunar base that sustains crew members for months is not just a stepping stone toward Mars in the engineering sense. It is a biological laboratory that will teach us what sustained exposure to 1/6 of Earth's gravity does to the human body over timescales we've never studied. A Mars mission that sends crew members to the surface and brings them back will not just be an exploration milestone. It will be the most extensive test of human biology outside Earth's environment ever conducted, generating data that future missions absolutely depend on. The temptation to skip steps, to go straight from low Earth orbit to interstellar ambition, is understandable. The destinations are more interesting. The engineering challenges are more dramatic. The short missions feel boring compared to the grand vision. But the biology doesn't care about the grand vision. It cares about the conditions it's in. And the only way to know what those conditions do over timescales no one has yet studied is to study them, step by step, in real human beings, in real space environments. There are no shortcuts through the biology. The question of who should go on such missions, if and when the biology is sufficiently understood and addressed, is also worth serious thought. Current astronaut selection is a process of finding extraordinary individuals, highly educated, highly trained, psychologically stable, physically fit, medically robust, with specialized skills that match mission requirements. The pool is small. The selection is rigorous. The training is years long. For missions of the duration we're discussing, the selection problem is qualitatively different. A 6-month ISS mission and a 40-year interstellar voyage are not just quantitatively different in duration. They require different things from the people who go on them. A 6-month ISS crew needs the skills and resilience to manage a demanding but finite professional assignment. A 40-year interstellar crew needs everything that a 6-month crew needs, plus the psychological architecture to sustain motivation and social cohesion and professional competence across 40 years with no return date, no news from home in any meaningful time frame, and no possibility of leaving if the choice turns out to have been a mistake. The screening tools needed to identify people who can do that don't exist, because the situation has never existed. You can select for psychological stability in environments we've studied. You can select for resilience based on how people have responded to previous stresses. You can try to identify the personality traits and coping styles most likely to sustain functioning across long-duration isolation. But there is no test for 40 years of confinement, because no one has ever been through 40 years of confinement. The first interstellar crew will be making a leap into a psychological unknown that no selection process can fully prepare them for. Some of them will thrive. Some of them will not. And the mission's planners will have to decide whether the probability that some crew members will fail psychologically is an acceptable mission risk, and what provisions will be in place to manage that when it happens. The ethical dimensions of this selection problem extend beyond the individuals selected. They include the families the crew leaves behind, the society that bears the cost of the mission, the people at the destination who may interact with what the mission produces, and the crew members themselves who must consent to an experience whose actual psychological and physical demands cannot be fully disclosed because they have never been directly observed. Informed consent for the first interstellar mission is, in a meaningful sense, impossible. You cannot fully inform someone about an experience that no one has ever had. You can describe what is predicted. You can extrapolate from what is known. but the gap between prediction and the lived reality of 40 years in deep space will be crossed only by the people who go, and they will cross it without the ability to change their minds after they've left. There's a version of this problem that can be framed as existential, and it's worth naming directly. The human species evolved on one planet. It has never left one planet in any permanent or biologically sustained sense. The excursions to the moon lasted days. The missions to the ISS are temporary deployments to a station that is fundamentally dependent on Earth for resupply and crew rotation. There has never been a self-sustaining human presence anywhere other than Earth. Everything we are, biologically and culturally, is the product of this one place. The drive to become a multi-world species, to establish humanity beyond Earth as a form of existential insurance against catastrophic threats to this one planet, is compelling in its logic. A species that exists on only one world is vulnerable in a way that a species spread across multiple worlds is not. If something happened to Earth, an asteroid impact, a runaway climate catastrophe, a biological event of sufficient scope, and humanity existed only on Earth, that would be the end. Spreading across other worlds provides redundancy. Insurance. The logic is sound. The practical challenge of doing it is where the biology enters. Because you can't establish a self-sustaining human presence somewhere else without sending human bodies there, keeping them alive and healthy there, sustaining them there without ongoing support from Earth, allowing them to reproduce and raise children there, and building communities there that can persist independently. Every step of that process runs into the biology. The physical health of the colonists. The reproductive viability of humans in reduced gravity. The developmental biology of children born in environments far from Earth. The psychological sustainability of isolated communities. The medical self-sufficiency of settlements with no access to Earth's medical infrastructure. The biological barriers to interstellar travel are not separate from the existential argument for becoming a multi-world species. They are the central challenge of it. You cannot become a spacefaring civilization by building better ships alone. You have to solve the human side. And the human side is the hard part. Not because space is trying to stop us, because we are what we are. Products of this world, dependent on it in ways that go to the cellular level. And the work of becoming something that can live beyond it is the deepest and most demanding scientific project our species has ever faced. One field that rarely enters the public conversation about spaceflight barriers, but that space medicine researchers take very seriously, is nephrology, the biology of the kidneys. The kidneys are the body's primary filtration and chemical regulation system. They regulate blood pressure, electrolyte balance, acid-base chemistry, fluid volume, and the excretion of metabolic waste products. In space, every one of these regulatory functions is challenged. The fluid shift toward the upper body changes. The volume and pressure signals the kidneys use to regulate fluid balance. The reduction in blood volume changes the kidney's operating conditions. The changes in dietary intake and metabolic chemistry that occur in space alter the chemical load the kidneys must process. The bone loss that releases large quantities of calcium into the bloodstream increases the calcium load on the kidneys, raising the risk of kidney stone formation. Kidney stones are one of the most painful medical conditions a person can experience. And in space, where there is no urologist, no lithotripsy equipment, and no surgical capability to remove a stone that doesn't pass on its own, They represent a potentially mission-ending medical event. Kidney stone risk for astronauts is elevated compared to the ground-based population. The combination of elevated urinary calcium from bone resorption, altered fluid dynamics, and limited water intake in space creates conditions that favor stone formation. A crew member who develops a large kidney stone on an interstellar voyage and cannot pass it faces a situation that, on Earth, would be handled with straightforward medical intervention. In space, the options are pain management and hoping for spontaneous passage. If that fails, the medical outcome is determined by what medications are available, which may include little beyond analgesics, and the willingness and ability of the crew member to continue functioning through severe pain. Dietary interventions that reduce stone risk are known and can be implemented. Adequate hydration reduces stone formation. Certain dietary modifications reduce urinary calcium and oxalate. But in a long-duration mission where dietary options are constrained, maintaining ideal renal stone prevention protocols across decades is not guaranteed. And the kidney's regulation of fluid and electrolyte balance is fundamental to the function of virtually every other organ system. A kidney that's operating under the altered conditions of long-duration spaceflight in microgravity, with its changed blood volume, changed fluid distribution, changed dietary inputs, is an organ working outside its calibrated parameters. The long-term consequences of that for kidney function over decades are, once again, not well studied. Because no one has studied a human kidney in those conditions over decades. The musculoskeletal system and the cardiovascular system interact in a way that makes the combined problem worse than either alone. When the cardiovascular system deconditions in microgravity, reduced blood volume, weaker ventricular muscle, altered pressure regulation, it becomes less capable of delivering oxygen to working tissues at high rates. The cardiovascular ceiling on exercise intensity drops. The maximum sustainable physical work rate decreases. But exercise is the primary countermeasure for bone and muscle loss. A cardiovascular system that can't sustain high-intensity exercise means a crew that can't do the exercise needed to protect their skeletal and muscular systems. The countermeasure is compromised by the very same environment it's meant to address. This is the kind of interaction that makes the biological problem of spaceflight harder than any individual component would suggest. The body is an integrated system. Change one part of it, and the changes propagate. The tools for addressing one problem interact with the tools for addressing another. The countermeasures for bone loss rely on exercise capacity that is limited by cardiovascular deconditioning. The cardiovascular countermeasures require cardiovascular exercise that is limited by the available exercise volume that the crew can perform without interfering with their work schedule. Maintaining a 2.5-hour daily exercise regimen across a 40-year voyage every single day, without interruption, without excuses, for decades, is not a program that fits naturally into any realistic mission design. People tire of exercise regimens even in the best conditions. Over decades, maintaining the discipline of a medically mandatory daily physical training program in the same space, with the same equipment, is a psychological challenge that layers on top of all the other psychological challenges of the mission. Countermeasure compliance is a documented problem even on the relatively short 6-month ISS missions. Crew members, under heavy work schedules, sometimes skip exercise. Sometimes equipment fails and isn't immediately repaired. Sometimes the pressure of other mission priorities crowds out the exercise time. Each missed session is a small increment of additional bone loss and muscle atrophy that doesn't recover. Over decades, the accumulated missed sessions could represent a substantial proportion of the total countermeasure effect. And there is currently no reliable alternative to exercise as the primary countermeasure. Pharmacological approaches can supplement but not replace the mechanical stimulus that exercise provides to bone and muscle. A mission that depends on a crew maintaining perfect exercise compliance across decades is a mission that is betting on human discipline being more reliable than it has historically been in any other. Context. All of these individual systems, the skeleton, the muscles, the cardiovascular system, the kidneys, the immune system, the visual apparatus, the neurological architecture, the circadian machinery, the microbiome, the reproductive system, they are not separate machines bolted together. They are a single, integrated biological system. A system whose parts communicate through hormones, through nerve signals, through mechanical forces, through the shared chemistry of the bloodstream. What happens to one part is not isolated from what happens to the others. The body doesn't experience the space environment as a set of discrete challenges. It experiences it as a total environmental change. And it responds to that total change as an integrated whole. The systemic response to microgravity includes simultaneous adaptations in the cardiovascular system, the musculoskeletal system, the fluid regulation system, the immune system, and gene expression across multiple tissues. These adaptations occur together. They influence each other. The cardiovascular changes affect fluid distribution, which affects intracranial pressure, which affects vision. The immune changes affect inflammatory signaling, which affects tissue maintenance across multiple organ systems. The altered gene expression affects cellular behavior in ways that we are only beginning to map, treating the biological challenges of spaceflight as a list of individual problems to be addressed one by one, in isolation, by separate research programs, with separate interventions, may miss the most important dynamics. The body is a system. The space environment challenges it as a system. The solutions may need to be systemic as well. Not just a drug for bone loss here, and a drug for immune function there, and an exercise protocol for cardiovascular deconditioning, and a lighting system for circadian rhythm, but a comprehensive understanding of how the body functions as an integrated whole in the space environment, and interventions designed to maintain systemic health rather than addressing each subsystem in turn. That kind of understanding requires a different level of biological knowledge than we currently have. It requires modeling the body not as a collection of organs, but as a dynamic network of interacting systems responding adaptively to its environment. It requires the kind of systems biology that has become possible with modern genomics and proteomics and metabolomics tools applied specifically to the spaceflight context. This work is being done, not at the scale the problem requires, but it is being done, and the direction it points is toward a more complete understanding of what the human body is and what it needs than anything the history of medicine has previously demanded. The biology of interstellar travel may turn out to be one of the most powerful drivers of biological understanding in the history of science. Not because the goal is to understand the body, but because the goal of reaching the stars requires understanding the body so thoroughly that the understanding becomes its own profound scientific achievement. The last thing to say is this. Every biological system we've discussed as a barrier to interstellar exploration is also a testament to the extraordinary complexity of what life has achieved on this planet. The fact that your skeleton dynamically maintains itself in response to mechanical loading is not a simple thing. It is the product of billions of years of evolutionary refinement operating on a system of biochemical signaling of extraordinary sophistication. The fact that your cardiovascular system adapts its architecture to the demands placed on it, optimizing its performance for its actual environment, is not a flaw. It is exactly the kind of adaptive intelligence that makes biological systems so much more capable than anything we build. The fact that your circadian clock runs in every cell, synchronized and coordinated across the entire body, maintaining your temporal organization across a lifetime, that is not a weakness. It is one of the most elegant mechanisms in all of biology. The problems these systems create in space are not failures. They are the consequence of taking a system optimized for one environment and placing it in a radically different one. The system is doing exactly what it was designed to do. The problem is the mismatch between the design and the destination. And that is solvable in principle, not by making the systems fail more tolerantly, by understanding them deeply enough to extend their operating range, by identifying which specific parameters they respond to and finding ways to provide those parameters even in an environment that doesn't naturally offer them, by developing the biological tools to protect what the environment attacks, by eventually, perhaps, changing the systems themselves in ways that expand the range of environments they can sustain us in. This is not science fiction. It is the trajectory that biomedical research is on. The destination is not yet in sight, but the direction is clear, and the distance between here and there, while enormous, is finite. The next time you look up at the night sky and find a point of light that you know is a star, not a planet, not a satellite, an actual star, let that image sit with everything you've learned tonight. The ship that could reach that star, if it ever gets built, will carry people, real people, with skeletons that lose bone in microgravity and cardiovascular systems that adapt to weightlessness and visual systems that change under the pressure of redistributed fluid and immune systems that let latent viruses surface, and brains that accumulate radiation damage and minds that carry the weight of years in confinement with nowhere to go. The starship of science fiction doesn't have to contend with any of that. The one that would actually work, the real one, built from real materials and carrying real human beings, has to solve every single one of those problems. Has to solve them, not partially. Has to solve them well enough that the crew arrives, after however many decades, still capable of doing what they were sent to do. That is the challenge. Not the distance. Not the fuel. The body. The living, breathing, bone-losing, radiation-absorbing, circadian-disrupted, psychologically burdened body that is every human being who has ever looked up at the stars and wondered. We are the ultimate barrier to our own greatest dream, and the only way through is to understand that more honestly, more precisely, and more rigorously than we have so far. Thank you for staying with me through all of this. If it changed something in how you think about space exploration, if it shifted the frame from the distances and the engines to the body that has to survive them, then the time was well spent. The stars aren't waiting for us to build the right rocket. They are waiting for us to become capable of surviving what lies between us and them. And the first step toward that is knowing exactly what we're up against. Good night.